if you worry about the exam, okay, I mean, which you worry at rest. I understand that this is a lot of information within a short period of time, at least for some of you who has a very limited exposure or experience in analyzing brain signals there. So, and I know that you don't have much time to prepare, so everything is kind of crammed there. But we have to still have to conduct an exam, and actually it's good for you if you take it. And so from realistic point of view, so here we are not somehow looking for an in-depth knowledge there within this short period of time. So you're primarily looking for whether you kind of grasp the principal concepts and some primary information, not about all possible details about some various papers or other methods we discussed. So anyways, I just prepare a small presentation to explain how, I mean, what we expect from you and what you expect from the exam there. And naturally, I would be very happy to talk to afterwards if you have any further query. So it's a 1 hour exam, it's because it's 2 credits. Total marks 100. There will be 2 types of question. One is multiple choice. There will be 10 of them, and each multiple choice question carries 3 marks, and there will be 7 short questions, which each carries 10 marks. So, in the initial lectures, I gave some samples, so I also added a few more there of each of the types. So, in the multiple choice question, each question will have 4 choices there. So, naturally, as it suggests, only one of them would be the most appropriate answer in that context. And as you know, often the MCQ you have to read really question carefully there. And even if you are not entirely sure about a particular option, there may be something don't exactly, I mean, not entirely certain, but still we encourage you to make an educated guess. It's not a random guess, I hope not. So, because we don't have any, I mean, usually whenever we set MCQ, we always also set kind of negative marking, but not here. There. So, I expect that you should not leave it blank, at least you make a reasonably educated guess. So, if you start, I mean, you will see, eliminate the options which are least likely, then often you end up on the right answer. So, okay, so some sample multiple choice questions. So, from the beginning, let's see, refresh our memory there. So, choose the incorrect statement about action potential. If you remember, that's, I think, the second lecture we talk about action potential there. So, action potential is all or none. Profiles of action potentials of one neuron vary greatly depending on the state of neuron. Action potential propagates in a non-decremental fashion, and the duration is 1 millisecond. Which one you think the incorrect statement? Incorrect. So, that's the point. You have to read carefully. If you that's where often the twist in MCQ. So, which one you think? A, B, C, D. Which one? Who gets the loud kind of strongest shout? C, I heard. Okay. D, I heard. Or poor A and B. Okay. Actually, if you go back to C, an action potential propagates in a non-decremental fashion. It's a correct statement. No. So, you have to choose the incorrect one, and also the duration is around 1 millisecond. So, that's also a correct statement. So, here the right option is the B, because the profile of an action potential of a neuron is always the same. It doesn't depend on the state of the neuron. So, it's just like kind of shape of the action potential is like a face of a neuron. Like you cannot change your face. You can put a little bit makeup here and there, but you can't change. So, that's similarly like a kind of neuron. So, the profile of the action potential is a kind of quite unique. It doesn't change depending on what state the neuron is. Okay. So, next about the EEG, which is slightly, hopefully, less confusing. EEG reflects summation of action potential, postsynaptic potentials, both action potentials and postsynaptic potentials, or none of the above. C, B, B. Okay, I can hear the B. Okay, some C telling, some C there. Anyone favoring C? No. So because I heard C, actually the answer is B, because EEG, okay, from the scalp surface, EEG cannot capture anything of action potential because it's too far to pick up action potential. It primarily reflects the summation of the postsynaptic potential. The next one is much easier for you. For an EEG signal with a maximum frequency of 70 Hz, aliasing occurs sampling frequency 256 Hz. D. So you can see I told you it will be straightforward. Okay. Okay, another sample question. So this is from ERP. So in ERP analysis, suppose you have ERP, if there is no clear ERP component, which of the following measures are more appropriate? Peak to peak amplitude, onset latency, peak amplitude, or the mean amplitude? Mean amplitude. Any other options there? So if you have any query about this question, feel free to ask, because you know ERP. Okay, I mean, yes, that's right. The mean amplitude is the right answer here, the right option, because if there is no clear ERP component, means there is no clear peak there, peak or trough, either positive peak or negative peak. So you cannot use peak to peak amplitude. So you can see you can eliminate the option even if you're not sure. Onset latency is something related to the peak. If there is no peak, what is the point of doing latency? Again, you can eliminate option B. Peak amplitude, there is no clear ERP peak there. So what remains? That's D there. Okay, so this one also should be quite straightforward to you. Regarding Welsh periodogram, which one of the following statements is incorrect? It smoothens the power spectrum estimation, it reduces the variability, increases the frequency resolution, allows calculation of a confidence interval. C. So you see, it's a pretty there is very little confusion. Yes, okay, it actually does smooth, it does reduce the variability, it does allow calculation confidence interval, but it doesn't increase frequency resolution there. Okay, some little bit later lectures. Suppose you want to, okay, it's very easy now. This is for tracking time varying content of constituent frequency of EEG signals. So this is primarily to check whether you are asleep in the lecture or not, because this is so straightforward. Which of the following method would be most appropriate? Fourier transform, principal component, wavelet transform, or independent component? C, wavelet. Anyone has any other ideas there? Because this is, I mean, to be honest, okay, even if you're just present for a few minutes in that particular lecture, you can tell that in answer that in fraction of a second. Okay, so now about ICA. So regarding independent component analysis, ICA, identify the incorrect statement. ICA is closely related to principal component. For a data set with n number of channels, you have square root number of independent components. ICA is effective in removing eye blink artifacts, essentially a linear decomposition method. B. So if you are not again very sure about it, can't remember, again you can apply the process of elimination. If you remember, when the ICA we talk about, it is before nonlinear lecture, so it is a linear method. So you can, and we talk about just after pre-processing. If you remember what context we talked about, we talk about in the context of artifact there. So the C is also the right options. And independent component, okay, even if you have no idea what it is, independent component analysis, principal component analysis, just the name suggests they should be related. Okay, even if you have no idea anything. So what it remains, that's a B there. So that's called, we call it educated guess. Okay. Sorry, this is something. Okay, now about some short questions there, which carries 10 points, 10 marks, but we really want you to just to be very brief there. So it's we don't expect more than 3 or 5 more than 5 sentences. Any answer there. Shorter the better, as long as you can highlight the main points, should be all right. Should be brief and to the point. Okay, so the first one, what are the evoked and induced brain oscillations? We talked a lot about that. So what would be the answer? What are evoked and induced brain oscillations? Anyone? Yes and no. I would give you benefit of doubt, but okay, but that's not the exact the definition, or not the right definition. Anyone? Okay. That's where it comes. So evoked oscillations have a strict phase relationship with an event. Induced, they do not. Okay. If you say that, just one sentence, you get 10 marks. Okay, so next one, what are the 4 assumptions in an ERP model? There. Remember? Start from assumptions 1. Let's see. If you remember, what is an ERP? What are the primary assumptions before that an ERP would be generated there, or before you expect an ERP to be present? Okay, that's assumptions 1. So ERP is invariant across repetition of trial. That's not an assumption. You cannot have an assumption about the number of trials, because that, though if you have an assumption, that assumption would be often violated, and or you cannot have a number as an assumption there.
Ongoing activity and ERP they are independent.
That is good, very good point. That's ongoing activities and ERPs, they are not correlated there. That's also very good point. Ongoing activities are random, they fluctuate from trial to trial there. And the 4th one is, which is related to the earlier one, if you look into the slides, they are linear superposition. ERP is just linearly added on the ongoing activity. That's it. Okay, so a little bit later lecture. Suppose consider an EG time series. We talk about was it yesterday? Yes. N data points, and you will perform a time delay embedding xt minus 3 t minus it should be tau. Okay, t minus tau, t minus 3 tau, so forth. What are the parameters d and the tau known as? What do you call d? Embedding dimensions. You just have to remember the name. Yeah. And what are the t? Or tau? Sorry, it's not the t. Time delay, right? Okay. And now list any 3 invariant measures that can be used to characterize the reconstructed state space. Okay. Entropy, exponent, very good. Correlation, dimension. Okay, that's it. 10 marks. Okay, so another conceptual question: why is the inverse problem in the EG MEG source reconstruction so difficult to solve? Because of that's right, there is no one unique solutions there. There are infinite number of solutions possible for the same field distribution there. Okay, 1 sentence there, not even 3 sentences. What is the peak to peak ERP amplitude measure? So you tell, just kind of measuring 2 neighboring peaks. What are its principal advantages and disadvantages? There. Do you remember ERP peak there? Any, I just asking any. You have to just okay. You don't have to remember exactly.
How much positive peak it means, how much negative
peak? You can say so, but that's usually not a very okay. I mean, I would not say that's a very appropriate criticism. There. You can say, think about, suppose, okay, a peak. Even if you don't forget everything, but a peak, calculation is always sensitive to noise. You're looking at only one point. So it should be, performance would be quite poor against noise. It's a disadvantage. Many other disadvantage. If you are measuring also only one peak, it's usually a nonlinear measure. Difficult to compare. Okay. How do you decide now? That's a slightly more conceptual kind of question. How do you decide about the number of wavelet cycles for analyzing TFR with complex modulate wavelet? I mean, we talked about it in quite considerable details. Yes, that's a very good point. There. because if you want some effect to be very short lasting, we use a fewer cycles there, or depending also or which frequencies we want to consider there. If you are looking for a lower frequencies, the number of cycle is less. If you are expecting effects in the higher frequency, number of cycles would be more. So in order to have an optimized, one can have a kind of graded variation of the number of cycles. Okay, so I put also some kind of FAQ there. Naturally, you might have more, but just first one actually is the best from the students from your point of view. So you can say, can I skip any lecture for the exam? There. I mean, I like to always say no, but here I can tell you yes, you can. There, because there will not be any question from tomorrow's lecture. Whatever we talk about or different, primarily, although we not talk too much. I mean, not giving too many slides, but there will be some about brain stimulation. Other there will be nothing, and also there will not be any lecture, any question from today's second half. Just because you're pretty close to the exam period, so we do not want you suddenly to. I mean, we want primarily to see how much you have gathered over the time. I mean, not at the very close to the exam. So yes, you can skip for the exam. I mean, for the exam part, but not for understanding part. So you have to remember this is about everything about the exam. Do you have to remember an equation? We talked about a lot of different methods there. No, you don't need to remember absolutely no equation there. It's counterproductive to ask you to remember an equation there. If you need, you can always check. It's mainly about the concepts that we are you would be examining. We somehow gave cited a lot of papers there. Often you might think, do you have to read all these papers? No, not for the exam. There, you don't need to read any of the papers. For scoring or answering for the exam there, but definitely if you want to have a much deeper understanding about the topics, which I hope you're aiming for, definitely you have to kind of read a lot of other related literatures there. I mean, I just gave you slides, but again all the slides are kind of distilled version of hundreds of papers there. So naturally you have to read the articles, latest articles, which way the field is progressing there. I just give you the background, kind of initial impetus there. For the exam, no. Calculator, no, you don't need to know calculator. Definitely, that's okay. Do you need to remember the details of a data analysis method? There we talk, because it's a kind of methodologically heavy course. There we talk at different methods. No, you don't need to remember the details. Only you have to remember just a simple words, as long as you can describe a method in simple words. If I ask you what is ICA good for, what it does, that if you can write in one sentence, it just separates the signal into an independent components, that would be enough. You don't need to remember how the ICA is computed, about the matrix, inverse, no. So there is no necessity to try to remember the intricate details of any methods. You will not be asked how the scaling exponent is computed, what is the steps of DFA, or what are the steps of other methods. There I talked about different methods, index of association, nothing. Those are just to kind of arouse your interest, and also to give you some ideas that you also can develop your own method as well. Some of the methods are intentionally introduced just for the fun, and also to show you that there are scopes so that you can develop your own method as well. So we are not testing here how much you remember the details of these methods there. Only you need to demonstrate that you have understood the general concept, what this method is good for there. As long as you can describe in a simple words, that would just be fine. Second, we also talk about a lot of applications of different methods there. So not just the methods, because we gave a lot of examples there. And naturally all these methods are applied in the different applied settings there, and naturally all the results vary from one paper to another paper. Do you have to remember? No, you don't need to remember any of the details there, whether some paper talked about DFA applied to the genetic data, or no, you don't need to remember. Those are again presented just to demonstrate the effectiveness of method and some what it can do there. So you will not be asked about any particular result about any particular method there. Some general there. So what I say is good for, what DFA is good for, what wavelet is good for, what independent component is good for, when you can apply, when you cannot apply. You will not be asked, oh, this particular paper they applied, what did they find? No. So it is again we are looking for primarily your understanding of the concepts, and suppose you have a research question, you know where to look at. So you want to see whether you have this kind of basic understanding of how to approach the data, and whether you have a reasonable understanding about the underlying concept. Finally, best of luck. Any queries or anything I missed? I'm there. So I tried to kind of prepare this so that I know. Okay, I mean, because of so much of information, often you feel that, oh, how can you prepare, start? But it is, as you can see. Okay, I mean, exam would be reasonably straightforward. There would be naturally some questions you have to see carefully, think carefully to choose the right options or think, but you will not be asked to reproduce and integrate details of any particular finding. No. Any question, anything, concern still I haven't covered in this kind of, everything clear? There. I hope you will take the exams. At least it's always good experience there, and also to see how much you remember. Okay, and as I mentioned, short answer type, just try to be brief there, exact to the point. Okay, so that's about the exam. Okay, so now to find okay, so today we talk about the brain connectivity analysis, which is the latest kind of trend in the field. So that's how we also kind of planned. So we started from pre-processing, then ERP, then time frequency, then nonlinear, and then finally how the different brain regions connect with each other, share information with each other. Just first, in order to understand that, whether they're sharing information or not, how we can quantify this interdependency. And the second is, because the brain regions are not, brain is a complex system, so how the different parts of the brain interdepend on each other, how we can characterize this. So that's the network theory there. So that's I will tell in the second half. For both cases, because these both are heavily mathematical, and both cases are the fields are, these are all, we're talking about different methods there. There are again many methods are there, and majority, or actually most, if not all of the methods, they are not developed in order to apply on the brain signal there. They're primarily developed, suppose synchronizations, you will see majority of the methods are developed in the context of physics there. And network theory, the concept was developed in 50s or 40s context of mathematics there. But what we found that although these methods are developed in a different branch, either in the physics or mathematical physics or statistical physics or math there, but still they are very useful to capture some essential properties of complex systems there, and diverse range of complex systems there. And the appeal is that although the different complex systems, they might be very different at the surface level, so how a brain works compared to how a society works there, because both are, or weather, how weather works, so both are all three are very different, right? But on the underlying, the hope is each of the systems, they share some of the properties of the organization of the system in a very similar way. So there the hope is that by applying this kind of tools which are developed in a completely different field, they might still capture some of the essence of these big, large, complex systems, which might look quite different from each other, but still they possibly governed by the similar underlying principle. So the first is today first half we'll talk about primarily on understanding or to investigating, to measuring the degree of interdependence between two signals or between two brain regions. So if you talk about the connectivity, there are three broad types of connectivity in the brain. One is the anatomical connectivity, one is functional connectivity, and the third is the effective connectivity. What is anatomical connectivity? So anatomical connectivity, it is kind of the set of physical or structural connectivity. So these brain regions or this group of neurons is physically has a synaptic connections with another group of neurons there, or another group of neural assemblies. So it's a physical structural connections there. And anatomical connectivity usually range over multiple scales, because if you consider at the very micro level, a neuron can connect to another neuron there. So that's a very micro. And you can also talk about neural assemblies. Okay, they also have a connections, okay, with another group of neural assemblies, or if you go a little bit higher level, say larger brain regions also connect with another large brain regions. And that's neurons. If I give you a simple explanations, and neurons usually considered the cell body of the neuron, they are the gray matter there, and the connections, they are the white matter there. So and if you study there are nowadays fantastic imaging technologies available to trace this white matter, or we call it the fibers, which linking one brain regions to another brain regions at the structural level there. That's by using diffusion tensor imaging. There you get a very nice fine-grained information about structural connection between certain brain regions with another brain regions there. So there are certain really almost like an information highway there. So some big bundle of fibers connecting one brain region to another brain regions. So those are structural connection. That's usually structural connections are relatively stable or static at shorter time scales. So as I am speaking, before and after finishing the sentence, structural changes, not much change. My brain structure remains similar, but it can also change over a longer period of time there through the brain plasticity there. So structural connections, they are very stable at a very short period of time there, but we know the brains change there. Structures change, and okay, I mean even in the adulthood, our brains can change, structurally can change. You can develop new neuronal connections there. If you're learning a new skill, you literally find that your brain undergoes structural reorganization there. So that's what we call neuronal plasticity there. So that's about the structure there. But when you look into the brain signal, so what we are measuring primarily, not really. If you have any queries, you can ask. I mean, I can tell whatever I know there. Okay. No, I'll not talk about plasticity that much, but it's a very interesting concept. A lot of these days the research has been going on to understand primarily the plasticity in the later stage of the development. We know that when we are young, the brain is plastic. There's lots of evidence there, a lot of different ways that the brain undergoes changes. But the recent research suggests that we do not actually lose this ability, or the brains don't lose this ability to reorganize there, both structurally and functionally there, even at the later stage of the life there. So now question is how we can characterize this there. Let me give you a simple example, which is often quite captured in the media there. I mean, we also done that. Suppose when you compare a group of musicians and non-musicians, we are also looking at the effect of plasticity. But one very catchy example, quite a few years back, showed that the researchers studying the brains of taxi drivers, especially in London. In London, taxi driver, London is a big city, a lot of lane streets and others complicated. And if you want to become a taxi driver, those days were before the GPS days. Okay. So they have to really have to memorize all the streets and there were a strict test before you want to taxi driver. You have to pass the test, which tell you, suppose you want to go from a certain street to another street, which route you going to take. So you have to mentally remember all these things. So if you want to have taxi driver, if you are working as a taxi driver in London, so you really have to have a very detailed map of London in your brain there. And what the researchers find, they did studying the structural changes in the brain. They found those taxi drivers working as a taxi driver, there certain parts in the hippocampus. Hippocampus is actually a brain region which is deep in the mid-temporal role. I mean, why is it called hippocampus? That's so kind of funny, because hippocampus in Latin means seahorse, because it's have you seen seahorse, the picture of seahorse? So hippocampus, the shape is also like a seahorse. Anyways, it is in the deeper brain structure, which is very important for memory and spatial navigation there. So what the researchers found, that these cab drivers, they have a certain part of the hippocampus is considerably larger than other matched controls. And not only on that, if you are working as a taxi driver for a longer period of time, then this enlargement is actually proportional to how many years you have been working on that. So it shows that it's not just brain changes and then it remains static, because the more you are working on, the more you are using all intricacies of the London maps there, so that it shows that the certain brain structure actually changes with respect to your experience and expertise there. This is a classic case of neuroplasticity. And there's another twist. Then later on, these same researchers go from that when they scan the brain of bus drivers. Now, they didn't find that much of enlargement. Now you see, why is it so? Now bus driver, they follow the same route. They are not finding a different route every time. They just have it. They're a route driver in London, just like Calcutta. They follow the same route every day. So there is nothing more new things to be learned. Once they learn, done. There. But the taxi driver, they always have to navigate different routes. There. So bus driver, they don't show this kind of a strong signature of neuroplasticity there. Or suppose, I mean, there is a lot of literature. So even if they found that, or take the case of another interesting example, musician there. So if you are string players, suppose sitarist or playing sitar or sarod, or in Western they did it violin and others, and they found that usually, you know, how I will control my fingers there is a representation in my brain there. So how each finger is represented, certain parts in the brain, they're allocated for processing all the control of individual finger. So you can have a clear cortical map. And they found that if you are a string player, then the amount of cortex that you allocate for controlling each finger is larger there, because simply for them finer movement is more important there. And through years of training, years of experience there, slowly your brain changes there, and then you need to allocate or larger brain regions there to control for the finer movement. That's also another case of neuroplasticity. There. Now question is at the expense of. Okay, there is another interesting question, because as you know, in the nature there is no free lunch. We call it economics. So there is nothing. So yes, there is no clear, I would say, evidence that all this neuroplasticity comes. Is there a fixed expense or is there a variable expense? In this particular the earlier example, suppose the hippocampus extension in the taxi driver, we do not know precisely whether it comes at the expense of any particular other brain regions. There is no clear evidence. For the musicians, yes, we know, but that's, I would say, is an indirect collateral damage, because usually if you are playing with your fingers, then they suffer from a particular neurological disease called dystonia as well as muscular dystrophy there. but that is, I would say, it is more like using our finger too much there. So it is, I would not say it's at the expense of or the cost of this reorganization there. But there will be definitely, whenever you are rewiring, structural changes means you are rewiring your brain there. So we know whenever you are rewiring, although you are trying to maximize your information processing ability in a certain directions, there will always be certain cost there. But we do not know yet if there is a fixed cost there. Okay, so that's about kind of neuroplasticity there. I mean, this is a very interesting topic, or another one which I given a very these days very interesting finding. You know, these days, I mean, this meditation is becoming very popular topic in the West in the research. Some of you are working. So there is a particular type of meditation training called mindfulness meditation there, and there is a special program called mindfulness meditation based stress reduction, which runs for 8 weeks there, which is very intensive. Every day you go meditate in a group, there follow a certain methods, it runs for 8 weeks. Although its main purpose is not self-realization. Its main purpose is primarily to improve your quality of life, to manage better stress and so forth. And what researchers have found that just by 8 weeks, so it's not a long enough time for the brain there to change, but brain does change structurally before and after there. I mean, now the debate is, we do not know whether these changes is really meditation plays a causal role or not, but at least we know even in a short span of 8 weeks there are structural changes take place in the brain there. Okay, so that's about the connectivity at the structural. Then the next branch connectivity is the functional connectivity. As I mentioned, any brain signal or any brain imaging data that we primarily gather, whether EEG, MEG or functional MRI, they are functional data there. So functional connectivity, what it does, it captures primarily some patterns of certain deviations from statistical independence there. So now you are using the kind of statistical sense there, and this often found between the distributed, often spatially remote brain regions, which is somehow related with how these brain regions are correlated, or whether they covary, or whether they are coupled by phase and so forth there. And this type of connectivity or interdependency between the brain regions farther apart, which is time dependent there, means there is a temporal structure in the interdependency there, and it's a fast temporal structure. Just like as I speak, the functional connectivity network is very different. I just stopped there. So it's just a couple of hundreds of milliseconds, but already the patterns of connectivity, functional connectivity would change there. And usually these patterns are model-free measures. Means you are not imposing a particular model. This has to follow a particular model-based correlation. So these are model-free there. So you are looking for some kind of statistical interdependence there, without any explicit reference to causal effects. And naturally, so this is now, if you can consider it's a mathematical problem. You have a signal X, you have a signal Y, preferably signal X coming from one brain region, signal Y coming from another brain region. How can you tell, even if they are not sub-brain region, how can you tell it's a mathematical problem X and Y are interdependent? Just like you can state it in a mathematical question, you can apply various methods there. Each of the methods can address some of the aspects or some facets of these aspects of connectivity there, because two systems or two signals can be connected in various ways. There is no only one unique way that two systems would share or show interdependency. So that's why there are different methodologies there for measuring brain activity to extract these connectivity patterns, functional connectivity patterns. So I will talk about today primarily about what are the different methods or the major types of different methods which capture this functional connectivity. And the third connectivity, which we call it an effective connectivity, this is also based on functional data, but here you are imposing a particular model, you are imposing a particular patterns, you are expecting this brain region should causally influence the another brain region there. So, you are imposing a certain causality, so certain direction of information flow from one brain region to another brain region there. Usually it is not model free there, so you are it requires specification of a specific causal model there, and you can infer this kind of effective connectivity in various ways, primarily through perturbations or also through some observations of temporal ordering of neural events. So, I will touch some methods there. And all these three different connectivities from the structural level, functional level and the causal level, they are not independent also from each other there. So, for example, as I mentioned, the functional connectivity, both functional connectivity and the effective connectivity are time dependent, and the statistical interactions between the brain regions, they do change very rapidly, and in what way they change, this reflects the participations of different subsets of brain regions there, also different pathways there. It depends on what task you are performing, it depends on what processes, brain processes are involved there, okay. And also it also change with the structural subsets that are relevant for learning and development there. Importantly, all this, as I mentioned, these three types of connectivity, they are mutually interrelated there. Naturally, if you consider structural connectivity should provide the major constraint on the kind of patterns of function and effective connectivity that can be generated there, because you expect in order for the system to elicit a functional connectivity pattern, there has to be certain structural connection, otherwise connection cannot emerge out of no structure there. Although the relation between structure and function is very complicated, there has been lots of studies aimed there, but there is no one unique answer. It's mainly problematic because there's a multiple scales involved there. The way structure and function related at the very micro level is very different from the way it happens at the mesoscale or the macro scale. Yeah. So, also the structural inputs and outputs of a given cortical region, it should act as a kind of, we say, connectional fingerprint there, and that should determine or should be a kind of principal factor of its functional connectivity patterns there. So, that is about tells you how the structure shapes functional and effective connectivity. The reverse is also true there, because functional connectivity can also contribute towards the final shaping of the underlying anatomical substrate there, either through directly or through activity dependent other different types of synaptic modifications there. Although naturally this process is slow, because structural connectivity is a rather slower process than functional connectivity. Functional connectivity happens at a much faster scale, but this can also influence the changes happening at the structural level, which is happening over a slower time scales there. So, this concept, this aspect of functional and effective connectivity is related to this concept of neuronal synchronization there, because there has been this aspect of neural synchronization somehow boosted by demonstration at the invasive level. That invasive level means when you record from single cell or group of cells, researchers found that these neuronal levels, when you have a micro recording, they are in tune with each other there. So, that leads to somehow the explosion of interest if at the micro level there is a cooperations there, and also how the micro level contributes to the activities at a macro level. Now, when you have the data, functional data obtained at the macro level, now naturally the question is how the different macro level signals are correlated or interdependent, and that's the kind of general form of, we call it functional integration there. So how it is defined, this neural synchrony, okay, this is tells you about it's a concerted interactions about neural populations or direct reciprocal exchange of signals between two populations, whereby the activity in one neural populations influences the second, such that the dynamics become entrained and mutually reinforcing. So if you remember, I highlighted at two particular points. This is reciprocal exchange and mutually reinforcing there. So means there is always kind of information exchange in both ways there.
So they have got feedback.
Yes, there.
How does the feedback actually controls
directly or indirectly? You cannot. You can talk about feedback. You can have a direct feedback. You also have a indirect feedback there. So I mean, in brain regions, there is no one in regions which act in isolations. So every brain regions or every neuron is connected with all of the other neurons. You do not find always immediate feedback forward and feed forward and feedback connections between two brain regions for all possible brain regions. Some pairs of regions, some pairs of neural assemblies you might find. Some you would find in a indirect way. Suppose, let me give you an example there. So, primary visual cortex, which is processing the elementary visual stimuli, and as you add a more complex visual stimuli there. So, what are the primary means? Just a line, edges, which are the primary visual attributes, and these are processed in the primary visual cortex there. And now you think about a shape of an object. That's a slightly more complex visual attributes, or a color there. So those higher order visual properties are processed in higher order visual areas. We call it the V1 is the primary, then V2, then V3, V4. V4 is for the color. Then there is a motion, which is in the region called MT there. So all these regions, they reasonably act as a kind of like a modular system there, primarily there. And then all this information is somehow initially hierarchically processed. Then finally, they at some higher, we call it multimodal brain regions there. So these are somehow integrated there, and there you get some kind of we call it the binding. I mean, at this moment, suppose here, if I look at this pen, this has a line there. So this is processed in the primary visual cortex. If I just give a kind of hand waving explanations, then there is also, I mean, edges, primary visual cortex. Then I'm looking at the shape, the color, which is processed in the V3, V4. There, I'm not moving it; it's a static. Then finally, it goes into higher order brain areas. Then all this, I get a kind of one unique experience that this is a pen. We call it the functional integration. So different brain regions which are processing rather different things, but somehow they mix together. But the point is also, now you will see a feedback. Although the primary visual cortex primarily sends signal to the higher visual areas, the multimodal areas, they also send projections to lower order, lower sensory regions there. So they might not always send, suppose V1 and V2, V2 may not immediately project back to V1 there, but V1 also get projections from higher order sensory areas there. So there is the kind of, you expect a certain aspect of feed forward or hierarchical processing, but also you have a global feedback there. So that's the idea. Otherwise, if you have every brain regions, if you expect, I mean anatomically, if you expect both their feed forward and feedback connections, there will be information processing would be slightly inefficient, because there is a certain advantage of modular processing there, and also you need the feedback, okay, for the information processing to be efficient there. So you do have reciprocal exchange, but in the very fine level, I mean from from the surface level, you always have an reciprocal exchange, because these brain regions are pretty large, but at the micro level, you do not always have. Suppose if a neuron A is connected with the neuron B, if you're going synaptic connections, it's not always neuron B also would feed back to neuron A. No, you don't find it always, no. But in the brain regions, if you talk about a larger kind of patch of cortical neocortex, then you'll find yes there. Okay, so now, as I mentioned, we talk about these different types of methods, how we can measure the statistical interdependency. Yes. Oh, the blind persons. Okay, what about in what sense? That's about another case of neuroplasticity there. So the blind person's brain is definitely quite different from a sighted brain. There are many ways they're different there. So that's a question of neuroplasticity, because a blind person, we were talking about visual cortex, right? So the blind person, what are they going to do with the visual cortex, because they don't have a vision, right? And the brain or nature doesn't like to waste resources there. Nature is usually quite efficient. So what happens in the blind person, if you're congenitally blind there, so other sensory processes like auditory processing, they start picking up the resources in the visual cortex, or other way around, visual cortex, our visual cortex in the same location is start processing sound in the blind. And that is the reason that, or that is one of the primary reasons, the blind person have a much acute sense of hearing compared to sighted person, because our brain, we are primarily visually dominated kind of species, so it's a large portion of our brain is visual cortex there. So if you don't use it, then it would be kind of a big waste. So it's an automatic if you are congenitally blind, the other sensory stimulus, primarily the audition there, is picked up by the visual cortex there. So that's why there's lots of studies which shows that if you're congenitally blind, you have much better sense of hearing, especially peripheral hearing there. Because usually, if something happening in front of us, some location of sound, we are very good in telling where the sound is coming from. But if something happening here or here, which is at the periphery, we are very poor, both in the vision and in the sound. But suppose if there is a one source here, another source here, so our spatial acuity in the both auditory sense, we cannot separate. So if you put a one speaker here, another speaker there, you cannot. Your brain would mix it up there. But presented to the blind person, there would be distinct different brain response, these two different spatially located speakers there. So they are much better there. I mean, this is just one example of auditory acuity, but they're much better in auditory processing there. Main reason is they have a more brain regions now picked up to process sound there. Okay, I don't know if you are asking about from that kind of perspective there. Okay, so there is a host of methods. Okay, I just possibly here 3, 6, 8, around 15 methods I listed here, but there are many more there. Methods which can give you some information about the interdependencies there between the two signals, because as I mentioned, this is a mathematical problem. You have a signal X, signal Y, you want to study if these two are related or not there. So this is a question, it is nothing to do with the brain. There is a simple mathematical question, and to be honest, the field of kind of statistics started with this question there. So I mean, long back, the Galton, Francis Galton, I don't know whether possibly you don't know, it's in the 19th century. So he was interested in various ways to study correlation. This is nothing but the correlation. You want to see whether these two are correlated. So he collecting data in a various contexts to study correlation between the two variables there. So he was interested in correlation between the gene and intelligence. I mean, before the gene means before the concept of DNA was introduced there, inheritance and intelligence. And also later on, okay, the famous mathematician, those days we didn't call statistician, Carl Pearson there, I think in early 20th century, 1904 I think, he introduced the concept of proper quantification, this correlation coefficient. We say Pearson's product movement correlation, that's developed by Carl Pearson there. So that's a starting point, but then later on, okay, lots of different methodologies have introduced in the time domain, frequency domain. Broadly, I divided into two parts, kind of two types there. So one case is you are expecting the correlation between the two signals are linear, or you expect the interdependency between the two signals is linear. So different methods like linear correlations or its frequency counterpart coherence or partial coherence, then more advanced the Granger causality or frequency domain as counterpart of Granger causality, partially directed coherence or directed transfer functions. So these are each captures one or certain facets of this statistical interdependency there, but they all can be grouped under a linear because they are all assuming this coupling between the two signals is linear. Now we know that the brain is a good candidate, strong candidate as in nonlinear systems there, and also there is no guarantee that the coupling would be linear there. So you can, or there is a kind of big list of other methods which do not require the assumption of linearity in the interdependency. So these are grouped under nonlinear methods, nonlinear correlations, okay, or general type of both linear and nonlinear correlation, that's under information theory, general higher order statistical interdependency, phase synchrony, which is very used, okay, in the brain rhythms because the phase is related to the oscillations. That's a good reason why phase synchrony is often the most preferred method in the EEG and MEEG, because we are interested in connectivity in a certain frequency band, certain oscillations, and oscillations, we know the phase is closely related. So there is a group of, again, subgroups of methods under phase synchrony, and then based on nonlinear dynamics, okay, where you do not have a precise hypothesis or expectation about a specific type of interdependency, you just assume a general nonlinear interdependency. We call it generalized synchrony. There is also a group of methods or index have been developed. So I will give you, because, and as I mentioned, this list is no exhaustive. I just listed those who are the primary methods there. Okay, so this is a kind of field which is growing for the last. Okay, I mean it started in late 90s there, okay, but then it reached its peak early 2000 or mid 2000 there, and then the network comes, because this is the one which is makes you enter to the network theory there. Before you will see, before you apply your network theory, you have to decide first how you want to find out whether the brain regions are at all interacting with each other. So I will just give you a brief overview of individual methods there. So naturally each method actually can be quite detailed, a lot of nuances are there, so I will try to avoid those there. I'll give you a brief overview, but before that I'll give you a break. Thank you. Okay, we'll come back. So as I mentioned to you, I'll give you kind of brief snippets of the major methods that we use in neuroscience. Although as I mentioned, none of them are actually, or except one or two, are introduced or developed in order to apply for neuroscience. Okay, so first is correlation coefficient. You have all studied long back. Okay, you have two signals, x and y. Okay, you want to calculate what is the correlation coefficient there. It's the same, it's a linear regression framework, and if you consider the absolute values, it's between 0 and 1. So if the 0 is means no signals are not correlated, 1 is a perfect correlations there. It has been dominant for many years because it was introduced in early 20th century there, because it is easy to compute by hand even before the invention of calculator there. And if we apply this correlation coefficient, suppose these are the two signals here. I showed you just an example here. So two pairs of signals, the green and the blue. So what you are primarily looking at, you plot the signals, make it scatter plot. If they're correlated, they will be along the line, all the points scattered. So first one you can see here, although they don't look like very strongly correlated. I mean, yeah, there is a similar kind of co-evaluation. You get a high correlations. The lower one there, although they look kind of similar, but because there is a certain delay, you can do zero min, yes. There. I mean, I don't know whether the signal was plotted here zero min or not, because just for the visual clarity, I just shifted a bit there. But I mean, this is just for the visual sake. I put it, kind of give it a DC. So the lower one you can see they look similar, but because if you look carefully, there's a slight delay between the peaks, and that leads to a very low correlation there. And then if you want to study, then you can do also time varying correlation as well. So that's what I was telling, that if you look carefully, this lower signal there, you will see the alignment or the peaks are not perfectly aligned there, and that leads to the low correlation. The advantage is correlation coefficient. It's a very yes.
It's not really, but then do you also look at time shifted?
You can do a lot of different versions of correlation coefficient there. So I'm not going into because correlation coefficient you can have a different you can look for the delays if there is, you can shift it, how much peaks are shifted. So it is very simple to calculate. It's easier to interpret, and there is a strong analytical support, so you can calculate the confidence limit there. But some disadvantages, you're only looking for linear coupling there, and there is also no direction on information there. So if the signal x and y are correlated, you cannot tell whether information flows from x to y or from y to x, and you need the signal to be stationary there. And most importantly, you really need this two signal to look very similar there. It is very unlikely if the two signals look very dissimilar, you get a high correlation there. In the EEG field or MEG field, there, I mean, for various reasons, you do not find using correlation coefficient anymore to study the functional independence, because it's notoriously unreliable for actual brain signal. But you will still find it's still very reliable in the fMRI literature there, when you have a voxels, the individual voxels there, and then you can study the correlations between the intensity of the voxels there. So the next kind of stop in these methods too is the magnitude squared coherence, which is nothing but same like correlation, but in the frequency domain there. So it's a frequency domain counterpart of correlation coefficient. Put it simply, you have the autospectra, the marginal spectra, and divided by the cross spectra divided by the product of the marginal spectra there. So you have a 2 x and y. So this is the upper one is the cross spectra here, and the lower one is the marginal spectra there. Again, there are different ways to get a better estimate of coherence. I'm not going into that. there. But if you want to summarize what kind of it is good for, or what are the boundary conditions, again it's very simple to calculate there. And this one become more popular after the introduction of FFT. You remember we talk about FFT, which makes the computation of power spectral density much easier there, because you have to compute here not only autospectra, but you have to compute the cross spectral power, cross spectral density. So after 1967 you will find suddenly a lot of interest in application of coherence to EEG, and also suddenly computer people, researchers could get access to. So that this coherence was used as a primary measure of functional interdependency almost for the next 30 years, from 1967 till late 90s. This was the main tool, principal tool to study functional dependency between EEG electrodes, or between MEG sensors, or between two brain signals there. That was the primary method. Simple to calculate, easy to interpret, and also like correlation you have a strong analytical support. You can have a confidence limit, and also you have a frequency related coupling information. That's very relevant for the brain, because you are interested in the oscillation specific interdependence. You want to find whether there is a stronger interdependence in the alpha band there compared to other frequency band there. So because coherence is a function of frequency there, so you can easily distinguish the contribution of individual frequency bands to the interdependency. But there are certain cautions there, because like correlation your signal needs to be stationary and also needs to be linear. If you do not have many trials or shorter durations, you have a poor estimation. It links to that, because in order to have a good estimation of a Fourier spectral periodogram, you need to have a longer data with a lot of epochs. That's what I do for averaging there. So naturally a coherence estimation would be quite poor if you only have a one trial. And also like correlation, it doesn't give you any directional information. It cannot tell you whether the signal X influences Y or the vice versa. And most importantly, it mixes amplitude and phase informations there. So it's the way it works actually, the phase information is completely lost, and that you will see afterwards. The phase information is very crucial in understanding neural interactions from the brain rhythm perspective. Then another method which has become very popular very recently, although it has been originally introduced in a very different field, in economics there. And actually this is one of the contributions made by Nobel laureate. It's called Granger causality. Clive Granger was an econometrician, so in 1969. So he proposed a very simple framework in the econometrics journal there, how to find causal directional flow between two interactive systems. Is based on a very intuitively very simple idea. Suppose Norbert Wiener, actually, who is a famous mathematician, the father figure of cybernetics from MIT. So in one of his commentaries on his books on cybernetics, actually he already introduced the notion of causality. He said past and present may cause the future, but the future cannot cause the past there. So means there is a role of time there. We call it the arrow of time, and role of the arrow of time is conceptually directly related to the causality there. So the Granger actually, what he did elegantly, he put this statement or this aspect in a mathematical framework, in a very simple autoregressive modeling framework. So what he proposed in his classical paper, which he got a Nobel Prize in economics later on there, so he proposed that you have a signal X, you predict the signal X by just information of its own past values there. Simple autoregressive framework. There you have a prediction error, and then you predict the same signal X by not only information of past values of his own past values, but also past values of signal y there. And if the second prediction is better than the first prediction, then you can say the y past values of y contain some informations about the system X. Then you can say the y has some causal influence on system X there. So that's a simple idea there. So that's what he introduced, this concept of predictive framework there. So if another system is really causing this another system y causing system X, then system y would contain some information about how the system X would evolve there, and the vice versa there. So he put it in the framework of autoregressive modeling, the simple linear autoregressive modeling. Then you study the prediction error. That's how your prediction performance was evaluated there. So it was proposed in, as I mentioned, in the field of economics. There's nothing mentioned of brain or whatever. It's just a simple mathematical framework. Then it was not known to the field neuroscience anyone, okay, because coherence was still the darling of the in functional connectivity methods there. Only in the very late 90s and early 2000, actually we also one of the first to put it Granger causality, but put it in the framework of state space. So one of the first in early 2000, then researchers started realizing the importance of a method which was developed or introduced in a completely different field could be quite informative in neuroscience there. So just give an earlier example here. So this is okay. So this is simple autoregressive framework. So here, so xk is expressed as a function of autoregressive certain past values of x there, and you have also you can express as a function of a certain past values of y and plus a prediction error, a noise, and you have this this individual weights autoregressive parameters there. Similarly, y can also be expressed as a function of the past values of x and past values of its own past values. So if xk depends crucially on the y, or means y causing x, then you expect a certain parameters, this b would be non-zero there. If they're all zero, all b's are zero, means the past values of y doesn't have any information.
You need to find out this AKs and these.
Yeah. So that's, you can do that, model order and others, okay, there. So that's definitely, you can follow the standard procedures for autoregressive process. But the concept is that you have a certain past weights, certain weights of the past values of another series will be non-zero there. Then you expect that okay, y would have a causal influence. And since this is a kind of a statistical framework of causality, we often say, and since it's introduced by the Granger, we use that called the Granger causes there. So just to make it's a kind of working definition of causality, so a certain aspect of causality. And if you see both are non-zero, both b and d, then there is a feedback relationship between x and y there. And you can also have similar, but in the information theoretical approaches, the same one. We actually, I didn't include it here. There is another branch has been developed to how to incorporate that in the information theoretic framework. So Granger causality, yes, it has now being very much used in EEG field, MEEG field, fMRI as well there, because we all are interested somehow to understand the directionality of information. All the statistical sense, but it can provide you some directional of information there. And advantages, you are not having any kind of, this is a stochastic formulation, so you are not assuming anything deterministic about the system. Some disadvantages, you are still assuming only linear coupling there, and the causality is somehow is not always very well defined, especially when you have bidirectional feedback or the bidirectional causality there. And if your data length are rather small, the short data samples, then it can lead to sometimes spurious detection of causality. And the fourth, as professor asked, you have to choose all this A, B, K, how many K before you go there. So, it is a parametric formulation. So, there is a certain decision to make the model parameter there, although there are standard rules are there depending on what kind of criteria you are going to use, the maximum information criteria or Schwartz criteria there, but still you have to choose a model order there, and that can be non-trivial sometimes. So, the related with a Granger causality, the Granger causality is a causality in the temporal domain. Now, in the brain oscillations you are interested in the frequency domain. So, how you can somehow use the similar framework in the frequency domain. So, partial directed coherence is almost like a frequency domain extension of Granger causality, but in a multivariate framework. So, here which is quite useful from another point of view, partial directed coherence, about the distinguishing between direct cause from indirect cause. What does it mean? Suppose you have a two system, 1 and 2, information goes from 1 to 2 there. So, there is no indirect no feedback. So, direct node 2 is receiving direct information directly from 1. Now, think about another possibility. 1 is influencing 2, 2 is influencing 3 there. So, here you can say yes, there is an information flows from 1 to 3, but there is no direct connection there. And sometimes we in the neuroscience, or often in the connectivity, where you want to find out whether there is a direct connectivity or information flows directly from one system to another system. Or another one, think about, okay, if you are looking for another configuration, there is system 1 is influencing both 2 and 3 there, but the 2 and 3 are not related. So, what happens if we apply pairwise analysis, means either correlation, coherence or Granger causality only between pairwise, then here in this case, yes, you get a good relation, proper relation for the Granger causality. But look at here, Granger causality would tell you also the system 1 is causally influencing system 3 there, but there is, as I mentioned, there is no direct connection. Or here, if you apply pairwise, then often you get these 2 and 3 are correlated, although there is no direct link. So, if you are interested in finding a direct or in distinguishing direct from indirect connectivity, the partial directed coherence is a good candidate there, or at least aiming to address that, because it takes multivariate data. It's applied not on the pairwise data, and it is, as I mentioned, it's a frequency specific measure. It's primarily applicable to linear, but with some modifications you can do nonlinear. As I already mentioned that. Okay. So, what the partial directed coherence, you just say similar in the AR framework, but it is a multivariate AR framework there. So, what you primarily do, you fit a multivariate AR model, and then you take the Fourier transform, okay, of the parameters, and then you just calculate in the matrix, the Fourier transform of the coefficient matrix, and then you define the partial directed coherence there. So, it will primarily get a matrix. So, the columns would give you the whether channel I, how it influences channel J as a function of the frequency. Give you a simple example. Suppose you have this very simple system that you are simulating. So, 1 is influencing 2, 2 is influencing 3, 3 is influencing 4. Suppose here, so there is a past values of x1, actually there is no element of x, no other x2, x3, x4, x5. So, x1 there is nothing. x2 also influences on the past values of x1. So, that is why you can see x2 is receiving input from x1. x3 is receiving past values of x2 as well as past values of x1. So, that is why the information flows from x1 as well as x2. x4 is, yes, receiving past values of x3, so only from here to 3 to 4. And x5 there is no x1, x2, x4, so it is isolated there. So, that is the simulations we, okay, I am showing it here. So, but you are just solving, you run these equations, you get the time series, you do not know the actual configurations how they are generated, and then you apply, suppose, partial directed coherence on this time series data. So, here you can see this is all these are frequency, normalized frequency here. So here it tells you about this individual column that tells you whether channel I or how much it influences channel J there. Forget about the diagonal, it is not important here. So here it shows there is a non-zero partial directed coherence values, a lot of peaks there from 1 to 2 as well as 1 to 3 there, but not from 1. You can see from each row it tells you how much it is being influenced by other channels there. So there is nothing here, so you can see other columns of the first row. So here it shows 1 influencing 2, influencing 3, it doesn't influence 4, doesn't influence 5. But if you look at the here, if you apply pairwise, if you do it compare 1 with 4, all pairwise would show information flows from 1 to 4. But here you are looking at it only direct information. Similarly, if you look into the second column, there's only here you find the second row and third row and second column. In second system is influencing third system, which is the close to true network. Similarly, for the third row, you will find the third system is influencing the fourth one is not influencing anyone, and fifth also is completely isolated there. So it shows that partial directed coherence, at least in this simple simulated network, it distinguishes direct connectivity from the indirect one, and the diagonal, as I mentioned, is not relevant. If you apply the coherence, which is the traditional method to study interdependency, and you will find quite an confusing results. Yes, one would show that 1 and 2 are related, 1 and 3 are related, but it also shows 1 and 4 are related, which is not wrong, to be honest, because 1 and 4 are indeed related there. But if you are interested to separate direct from indirect connectivity, then it is a wrong conclusion there. So similarly, you get quite a confusing network diagram, estimated network diagram, if we apply pairwise connections there. So PDC or the partial directed coherence, and there is another cousin, we call it the directed transfer functions there, just different formulations there. The advantage is that it is not applying only on the pairwise, it takes all the data, means you have the whole covariant structure in one framework. So it considers the structure of the whole data set there, and it has very nice can capture the directional influence there. It's reasonable because it's based on AR model, you can compute very fast there, and it provides the frequency related information about the directional. Disadvantage is mainly linear interaction, because this is linear AR modeling. I mean it is frame. You can introduce nonlinearity, but there are certain cost. And disadvantages, its analytical limit is very difficult to obtain, analytical confidence limit there. So it's a certain degree of arbitrariness there, where you put a cutoff if you have only one condition there, PDC. And also it's sensitive to certain preprocessing there. And again, like Granger causality, it's a parametric modeling, so you have to choose what would be your model parameters. Now, going from time and frequency domain, you can also apply the main vehicle of information theory, mutual information, because functional connectivity, as you know, it is just to measure any type of statistical interdependency, and mutual information, by the name suggests, it's a very good candidate to measure that any aspect type of functional interdependency there. So mutual information, if you have a one-dimensional, two-dimensional time series, you can calculate mutual informations there. Although, you think it's easier to calculate, but depending on your data, depending on your data properties, what algorithms you choose, that determine very much the sensitivity of your estimation. And especially mutual information is very sensitive. For high-dimensional data, very sensitive on the noise there, on the data size there. But the advantages of mutual information is it reveals both linear and nonlinear coupling, because it's just looking for statistical interdependency there. And because it is information theory, you have a strong theoretical background there, so you can interpret, you can come up with a good confidence limit, is a good analytical support is there for mutual information. Disadvantages, primary disadvantages there. So despite it is very powerful there, or theoretically powerful, I would not say statistically very powerful, theoretically powerful, but it's difficult to estimate for limited data there. And for the time or tasks related brain changes, which often the data are short, noisy, single trial level, mutual information estimation is notorious there. So, I mean, if you're interested, I didn't mention it there. We wrote a very long papers on different ways you can calculate mutual informations or conditional mutual informations to calculate the causality. I can put it, but that's a pretty mathematical. But it's important to remember that they are pretty noisy for high dimensional data, and there are different ways to deal with this the noises and as well as dimension. Huh?
How useful is this for neuroscience?
It is very useful there, but definitely depending on your data. If your data is if you're looking for short trial or short segment for single trial, then it is very unreliable. If you have a longer durations, provided the data are stationary there, then you get a reasonably reliable estimation. But mutual information is also you don't have a frequency information, but what you can do, then you can do bandpass filter the signal and then calculate mutual informations on individual individuals kind of brain oscillations. That's where you can somehow compensate there, but that's also not very good, because then you do not get a overall frequency band specific representation in one framework that you get in other methods there. So short answer is it is useful, long answer is it depends on where you want to use the quality of the data there. But certain aspects of mutual information or certain aspects of mutual, and its variance, because there is another group of methods based on information theory, we call it transfer entropy there, which are more used in the neuroscience there. Or you can use this, some aspect of conditional mutual information there, if you want to interested about the finding the delay between the in terms of transmission times there. There has been a few studies, including us, we also showed can be useful, but the computation is fraught with difficulties, reliable computation there. So that's, we mentioned the problem driven estimation for high dimensional system there, and also you don't get any frequency related information. So then there is another methods or which is called the phase synchrony. Again, these are all developed this is developed in the branch of physics, theoretical physics there. The concept of phase synchrony in the field of coupled oscillators there. So phase synchrony, classically it means is an equivalent to adjustment of frequencies. We know that in the phases are a crucial properties of oscillations, but here importantly here, so you are only interested in understanding or assuming an existence of a certain relationship between the phases. You don't care for amplitude there. And another aspect is the phase synchrony is found to be quite sensitive for nonlinear coupling or nonlinear systems, and also quite robust against internal noise there. So internal noise is very different from additive noise. So internal noise means this is which you call dynamic noise, when there is a certain noise inside the system there, and that makes it very difficult for majority of the other methods there. But phase synchrony actually quite thrives on internal noise there. You will see, I'll give you some simple kind of explanations. So how you can do phase synchrony there. So you have in stepwise, you have these two signals, say 2 EEG coming from two different electrodes or from sensors there, and you are interested in the phase synchrony in a particular frequency band. What EEG you have? The raw EEG, white, but you have to remember the phase as such is related with an oscillation. Phase is only meaningful when you talk in oscillations. And if you recall earlier, what is an oscillation? it is a narrow frequency band. If something has a wide frequency band covering wide range, we don't call it oscillations there. So phase is related to the oscillation. So if you calculate the phase of in this signal, you can calculate it, but it doesn't have physiological meaning there. If you apply, suppose, a Hilbert transform, you can calculate the phase there. People have done that, but those are not the right way to do that. It doesn't have any physiological meaning if you apply phase on the real data, real data which are wideband there. So whenever you talk about the phase, you have to make sure that you are addressing a narrow frequency band there. So means how to do that? You bandpass filter. And that you can choose which frequency band you are targeting, alpha band or theta band or whatever. So you filter each signal there, so you get the filtered version, and there you can calculate instantaneous phase of blue signal, the signal blue, and the signal red there. So that's what the instantaneous phase. I'm just putting it overlay on the top of each other there, and that you can do different ways. You can calculate instantaneous phase. Often the most used is the Hilbert transform, which is very easy to calculate. So now you can see here, so when you overlay on each other, just for the visual purpose I'm showing here, so there is kind of not clear similarities, but at certain point suddenly these two phases are in lock there. Means they have, here in this case, zero phase difference, but you also can phase difference when they maintain a constant phase lag there. So here it shows that, okay, this suddenly some part over the segment, the instantaneous phases of the two signals are the phase difference remain either here 0 or remain constant, and those are the segment of phase synchrony there. That's the idea, but that's in a very simplistic way there. But in reality, sorry, moment, I'll come back. I should come back to that. Just to show you here a very simple simulation, as the phases, okay, earlier I gave you an idea the coherence, which is mixes the amplitude and the phases, but the two oscillators can coupled in terms of phases, although their amplitudes might be completely uncorrelated. So here, just show you two coupled oscillators there. So 1 and 2 stands for oscillator 1 and oscillator 2 there. Simple nonlinear systems, there is a coupling coefficient is C there. So that's your coupling coefficients there, and then what you can do it here for different very weak coupling. One is a 0.01, one is a 0.027, one is the case of 0.035, three different coupling strength. And at all three coupling strength, if you plot the instantaneous amplitude of oscillator 1 and oscillator 2, in all three you will find no relation, because it fills the entire state space there. So there is no clear relation in the amplitude. But let's look at the phase difference series. So here I'm plotting the instantaneous phase difference between the two phases of two oscillators. So for the blue, when the coupling is really weak, the phase difference series do not converge, it just goes on and on. So it means there is no clear phase synchrony. Look at the red one here. So intermediate coupling, yes, they remain quite stationary for a long period of time, then there is a slight jump here, then another again they remain quite stable for quite a while, then there is again jump, and so forth. And for slightly stronger coupling, the phases remain phase difference remain constant. So you can talk about really, actually both cases you can talk about phase synchrony there. And also is to show you that the phases carry independent information, or information which is independent from the amplitude there.
If there are two oscillators.
Yes, this is actually simple two oscillators. Yeah.
Then
Simplistic.
phase is not ambiguous.
No, we are not talking about. This is talking about the phases of individual oscillators. So this is here we are plotting here. So phi 1, that's the instantaneous phases of the oscillator 1, and that phi 2 is instantaneous phases of the oscillator 2. So that's about, kind of show you that the phases are really worth investigating on their own there, and especially if you are looking into the kind of coupling between the brain oscillations. Now there are some interesting aspect. I showed you earlier the phases remain constant, but then there is a jump. And what happens if you try to understand when the two phases are two oscillators are somehow coupled, the phase coupled, so relative phase equations can be expressed that, okay, so this is mn coupling depending on their natural frequencies there and the coupling functions, and then and then there are some noises there which is inside the system. This noise is not an additive noise there. And what happens this inside the noise inside the system there? So if you want to model this, how the phase difference fluctuates between the two series, we usually is model in a kind of double well, we call it there, as like a ball here. So ball is either settled between, okay, there is a small kind of well here or another well here, and these two wells or these 2 points of stability is difference is 2 pi there. So what happens if the ball that represents the phase difference dynamics there. If the ball remains at this place, means the phase difference is constant. There is no change of phase difference. But this noise is all the time trying to destabilize the phase difference series there. If the noise is small, there it cannot destabilize. It's remain fluctuating, but remain within this stable region there. So then you get a relatively stable phase difference. But if the noise is slightly strong, then this ball hops from this point location to this location, and that we call phase slips there. And phase slips, these locations are usually separated by 2 pi. So that's the noise induced phase jump there. There has been an experimental verification and kind of mathematical foundation that's the case. So now what happens in the real system, this is about even in a kind of model system. In the real system, we know there is an internal noise there. So you do not find that the phase difference series rarely stays at a stable locations there. It hops from one location to another location most of the time there. So that's why there is a lot of phase slips, and these phase slips are often quite irregular in the actual neural data or actual any data there. So there you have your synchronization transition from one location to another location is very smear, and in order to capture that whether the phase difference remain within a certain angle or certain zone, you can only do that in a statistical sense there. And what you do not want these 2 pi phase jumps there. So that's why you calculate the instantaneous phases, and then you take a simple operation modulus 2 pi there. So you get rid of all the phase slips, and if you do that, so that's it shows in a slightly more complicated cases. So again, two coupled oscillators, there is a weak coupling, there is no coupling, the phase difference series you can see here, you can see phase difference series looks kind of very noisy there. It doesn't show this. Earlier I showed you, okay, kind of stable, because the noise is acting quite heavily on the oscillators to remain entrained there. But when you introduce a slight coupling, look at the phase difference series. This and these two doesn't look very different, right there. But then what you do here, you take this series and then take a modulus 2 pi operation. So you get rid of all the phase slips, and then you study the distribution of this one modulo 2 pi. Now you can see there is a very nice peak of the phase difference series. So it shows that although visually you don't see any clear kind of stable phase difference, but if you get rid of the noise slips and then study the distribution, then these two oscillators, they show their phase difference series, they show a strong preference towards a certain phase angle. So now means you have an idea that whenever this distribution of the generalized phase difference series, there is a peak, that would be a sign of a statistical evidence of phase synchrony in the real data there. So now you can measure how peaky the distribution is, because in this case, if the distribution is flat, then there is no statistical evidence of phase synchrony. If it goes the peakier the distribution, then there is a stronger evidence of
When you have two oscillators, which are coupled,
yes.
then they are going to be pulled.
That's right.
And
I mean, here you can do it both for identical and non-identical oscillators. So this is actually one here in this case identical, and you can also find the same thing for non-identical oscillators. I mean, their natural frequencies are different. There. So this also actually showed here. You can also get a phase coupling for a slightly non-identical oscillators. There means phase coupling is also not equated to frequency coupling. There. And most importantly, a very small coupling can introduce the phase correlations there. So means phase information is quite sensitive there, but you can only measure statistically there. So now you can apply a simple information theory measures how peaky your distribution is there. For the control cases, you expect a flat distribution, which is a maximal entropy there, and then you calculate how much this one with respect to a flat distribution. The higher the values normalized, the higher the degree of phase synchrony. So that's the method proposed quite a while back in the theoretical physics domain, then later on applied to the MEG, and lots application in the EEG. I mean, we applied 15, 16 years back. Now there are so this is just to show you the same one. So different cases, the phase difference series here. So here you can see there is a stronger statistical sense of phase synchrony, because as I mentioned, there is a stronger peak there. Here the peaky tendency is less, and this is more of an uniform distributions there. So you can quantify, I mean, although I'm showing you visually, you can quantify the peakiness of the distribution of the generalized phase difference series there. You can also measure the phases on a simple phasor unit circle there. So here you can use it, we call it based on circular variance. So you calculate the phases of individual oscillators or individual electrodes channels, and calculate the phase difference series at each point there, and you plot it on a unit circle there. If your entire duration of your recording, and you see where it's actually projected. If it's projected everywhere, all around the unit circle there, so naturally you will find that there is a very almost no phase synchrony there. But if you will see there, most of the time that it occupies a certain zone in the unit circle there. So then it suggests that again statistically the two time series, 2 EEG or MEG channels, they show a preferred phase relationship there. And if it's, and then you can calculate how much clustering it shows here there, and that you can do it by simple variance on the circular. So in these extreme cases, if it's all phase differences at all points distributed over the entire unit circle, then naturally the circular variance is the maximum. Here the circular variance is the least there, because the phase is showing more clustering at a certain segment of the unit circle, and here it is slightly in between. So if you can calculate 1 minus circular variance, then it will be higher the value, higher the degree of phase synchrony there. Phase synchrony, it is advantages is neurophysiologically very meaningful there analysis there, because obvious reasons the phase is related with oscillations. We are interested in oscillations there. So it is, as I mentioned, the most used in the last 15, 18 years there, and this is literally replaced the almost the coherence analysis there, and it's suitable both for nonlinear as well as non-stationary signal, because you can calculate, you can apply Hilbert transform, which is very good for dealing with non-stationary signals. So you can calculate phase different instantaneous phases application by applying Hilbert transform there. So it's suitable for non-stationary processes as well, and it's very sensitive to weak coupling. So if the systems are coupled weak, I showed you the phase, it is immediately reflected in the phase, and you have literally very limited assumptions there. So you can apply to your data quite easily. Only assumption is that, which is also disadvantage, is the phase is meaningful only for narrowband signal. So you should never apply, although you will find some papers which should not be published, but phase is only meaningful for narrowband signal, because phase coupled with oscillations, you have to make sure that it is it's somewhere narrowband, and the directional information is not clear. So, if a system or a causal or a signal X, electrode X and electrode Y showing phase synchrony, you do not have immediate information whether X is influencing Y or Y is influencing X there. Although there has been, after saying this, there are certain improved methodologies which can use a slightly more complicated way. You can calculate phase directional information by using phase. Quite a few actually methods are there. Each has certain sets of assumptions. I don't know whether I have included this. One is, just for the sake of completeness, as I mentioned, there is one is imaginary part of coherence there, and the related one, the phase lag index there. So these, both of these, another one which is I forgot the name, is introduced in the physics. They using considering development of they call protophase, and using protophase you can calculate the direction there. But I haven't, I mean, other than phase lag index, which can give you the direction information. I think some I possibly mentioned in the afternoon there, but based on certain assumptions they work well there, but they also have a certain limitations, because often those indices are very sensitive onto the lag there, because their direction is based on the lag. This is not an instantaneous causality, because often in the we know that although in the brain you expect that two signals would be somehow show some lag in their coupling, but on the surface recording you are unlikely to get a lag there. So it's an instant zero phase or instantaneous synchrony you are more likely to get if you have a recording from the surface, which is EEG and MEG. But nevertheless, I mean, altogether the phase synchrony is currently the most used technique in the EEG MEG field. fMRI also there, but fMRI, because correlation is more used, because it's very easy, straightforward, and there is a lot of lot of voxels to do there. Okay, I will stop it here. Then I will because we also have it today afternoon as well, right? So I'll continue in the afternoon directions. Okay. Any queries? Anything? Everything clear?
No references.
Huh? Oh, no references. Okay, there will be references if you're interested. Okay, I don't know whether you will be afternoon. By the way, there is a small corrections to my earlier. I said there will be no lecture, no questions from the afternoon lectures. But remember, afternoon means there will be no question from the network. There will be some, possibly there will be some about the synchrony, because synchrony I thought I will kind of finish it today. No reference. If you have references, kind of self promotion, although this is an old paper, but this is old paper of myself, long 10-year-old, 11-year-old, because long back I used to do this kind of analysis quite a while. This is a reasonably good overview. It is, thankfully, it still gives me kind of few hundred citations every few years. So yeah, it's a good starting point if you are interested about studying the kind of in a one paper broad overview, which is a 40-page paper. So that's possibly the reasons it gets 700, 800 citations. Okay, it's a good starting point as a coverage of different methods there. So if you're in, I mean, I avoided individual references there, so you can find a lot of cross references from here about the methodological aspects of different methods and its application to primarily to the brain rhythms there. So that's the reference, but there are definitely individual, because all these individual methods are proposed by the researchers from different community in different field there. Because as I mentioned, phase synchrony introduced by the group in Potsdam Institute of Physics. They're physicists, theoretical physicists. Mutual information, then suggested by completely information theory in 1949. Later on, partial directed coherence primarily introduced by a Brazilian group of researchers there, Bacala and Shameshima. Grandeur causality introduced in econometrics there. So you can see this is now kind of melting pot where the people with a different expertise coming from different community, and they develop the methods. But the beauty is the many of these methods, okay, they can be equally applicable to the neurophysiological signals, and that's where I think also the major kind of I would say beauty as well as an interest in the neuroscience there, because many of the things that we do in the neuroscience are somehow borrowed or inspired by the development in other fields. So it's a very interdisciplinary there. So and that possibly explains why I am never been taught as a psychologist. I'm in the Department of Psychology. So it's a good scope of contribution, and doesn't matter whichever field you are from, there is always a good scope of contribution or bringing fresh ideas. That's very helpful usually. We like that, the cognitive neuroscience, bringing people from different expertise, from different disciplines there. So where we have a common platform to talk about there. Okay? Heart rate variability from, I didn't talk about heart rate variability from EEG signal. I just showed you a figure where the method was introduced in the context of heart rate variability there.
Because there is a one disease you mentioned, that the heart failure.
Oh, that's a congestive heart failure. That's because that method.
Some of the cases the normal subject also within that heart rate variability. So is there any difficulty?
Is there a difficulty? Not really, because many of the things, as you know, although they were initially introduced in a different field, but if your task is primarily, suppose those particular example, this multifractal, that's what original paper was published in Nature in 1996, which shows that the heart rate variability in the congestive heart failure patients, they show impairment in multifractal. Okay. Now the question is, multifractal is a general phenomenon, and there they found some applications of or some impairment of certain multifractal aspects in a heart disease. That does not anyway mean that multifractal is intrinsically tied with hearts there. So now question is, that also doesn't mean that multifractal you also get an immediate evidence in the brain, but what it means, definitely all these methods that are developed in a different field there, if you apply them to a new field, provided that you are applying carefully, that provided all the assumptions are met there and you are having a judicious applications, then you are in a better place to, first of all, to characterize your system from a new angle, which may provide new insights that you might not get by other traditional methods there. That's the first aspect. And the second is, suppose if you are even approaching it from a very engineering perspective. So I want to use this classification. So I possibly do not make too much of or put too much importance what measure it actually measures, as long as it serves my purpose to classify different brain states. Suppose I gave this some preliminary efforts of using these multifractal based methods to categorize different sleep stages there. Now no one possibly would be claiming that whether there is really a severe change in the multifractal dynamics in the different sleep stages there. Even if it happens, there is still a long way to prove that, but nevertheless, from the signal processing point of view, if you can demonstrate that there is a certain features in your brain, that features derived by application on a different methods, here in this case multifractal, if that feature reliably distinguishes from one brain state to another brain state, that serves your immediate purpose of classification there. So there lies the kind of the two main interest in application or of all this diverse range of methods there, from both from linear analysis, nonlinear analysis, network theory, and all scaling analysis. Otherwise, if you think that you are only apply to the methods only relevant to the brain, then to be honest, you do not have much reason to go beyond ERP and just looking at amplitude there. Then your endeavor, everything would stop there, because ERP and just looking at amplitude where there is a peak, where there is a latency, that's it there. but then you do not have, then to be honest, then the branch of signal processing will not develop. But as a signal processing, these are complex signal there. You want to get a better characterization there. Suppose yesterday we talked about, someone asked about dimension, correlative dimension. So now we cannot prove that whether brain is low dimensionally chaotic or not, but applying these techniques, if it can help us to get a slightly better understanding about its underlying dynamics, and if it helps us to distinguishing between different brain states, to help us distinguishing between as used as a biomarkers, then very likely these advanced methodologies, although they come up with a lot of different assumptions, they might give you some interesting information about your underlying these complex systems that would be difficult to reveal by applying other traditional methods there. So that's why, yes, you have to be very careful whenever you are applying any methods, so otherwise all be garbage in and garbage out, we say. But once you've been careful about judicious applications there, and then you interpret your results within the boundary conditions that the method or the analysis provides, then it is a good adventure, and that's how the field actually evolves there. So you will see in the next half we'll talk about network theory, which is there in the mathematics in 30s and 40s there. But then suddenly there is a one paper which shows this application of network theory in a completely different. They actually studied application of network theory in studying how there's one particular example they did how the actors in the Hollywood are connected. So whether means if two actors are work in the same movie or not, and they found there is a certain interesting structure emerge out of that. And that's where I think the beauty of science, that's whenever you apply an idea or something and inspired by that, and then you apply on a different field, and then you get surprising outcome. And all the all the major discoveries are primarily made by this kind of accidental discoveries, or this. If you take a very routine path, then very soon you will reach a kind of dead end there. So that's the kind of the idea behind interdisciplinarity. So why we do need people with a different expertise from different disciplines, because idea is the same. Then when you bring people with a different expertise, then the overall outcome you would get, it would be greater than the individual contribution. And that's the idea of nonlinear system as well. The whole is greater than the sum of its parts there. So that's almost the ethos also of the neuroscience, because we know the signal is very complex, information rich there. So that somehow begets that looking for other methods which could be useful, but at the same time you are one should be very careful about application in the right way there, and also to remain within because each application has their own constraint, own assumptions. So you should be very careful about interpretations there. Okay.
Time frequency representation using wavelet. So let's say I blink. One is the I blink, and for the mother wavelet we prefer the dB6 or dB10.
Here I talked about, that's you are applying the wavelet in the scale multi-resolution functions, but here I showed it primarily talked about complex. This is not, we didn't have any scale version. This is a complex Morlet wavelet. Depending on what you are looking for using multi-resolution wavelet, that what purpose you want to use that. If you are, suppose the way I presented the wavelet in the time frequency, these are not good for reducing artifact. No, already artifact is done already there, and then you are applying wavelet to calculate how the oscillations change with time. And now what you are telling, that's primarily wavelet can be used as a filter, as a wavelet can be used as a noise filter. There, certain scales would be more relevant for or associated with a certain artifacts. There are certain pros and cons of that method, but the way we discussed wavelet in this lecture framework is primarily your data are clean. You are not using wavelet to get rid of any artifact. The data already clean, corrected, IC or whatever methods blink, there is nothing, and then you are looking at how the brain oscillations change as a function of time. So there is no mother wavelet, it's only one wavelet, you're just on a different time points you're using. So there is a one wavelet as a fixed, you are not changing any scale here, you are just using number of oscillations. So that's it.
I want to the frequency with respect to time oscillation. Like so, suppose say for example one I think with a 5-second duration. So I take a mother wavelet of a single I blink, and then like the flow over the signal, and then I got the 5-second duration. I got the frequency, and so in this way we choose the mother wave as a I blink.
Well, as I mentioned, there are certain methods using wavelet as a filtering noise reduction algorithm. So then you're using as a wavelet the different scaling that. But as I mentioned to you that, okay, I'm not telling anything against about that particular method of using wavelet in that, but here the way we use the wavelet, that's quite a different types of wavelet, because even a same, not same wave, because these are two different wavelets, can serve different functions there. Now what you are asking primarily that, okay, if I use a wavelet as a multi-resolution framework, then can I use it to reduce the eye blink there? Fine. Yes, you can, but I would not recommend that, because I would say if you want to reduce the eye blink, I think ICA would be much better performer there. So why do you complicate a situation? I'm not saying against that you should not invest, but I think if you want to correct for the eye blink there, actually there is a one method which is I'm aware of, that they use wavelet on the independent components there. So wavelet based ICA, in order to even improve the performance of the ICA based on wavelet, we also did instead of wavelet you can apply EMD on the independent components also to reduce, because if there is a problem called split variance, means if a one eye blink is split between two independent components, then, and if you reject both independent components, you are also reducing some neural signal. So you can clean individual independent component by applying wavelets. I know there is a paper using wavelet cleaning the independent components, or you can use EMD. Okay, yes, you can do that. But also there is other one method I know, using just a simple wavelet to reduce eye blink there. Yes, I have nothing against about that. So because the nature of the signal, there is hundreds and thousands of papers there, but what I'm trying to give you here kind of a streamlined approach. So I'm nothing against if you have found a one paper which addresses that problem in one framework. Yes, that if it works, fine. But if your primary aim is to remove eye blink, in our experiences ICA does reasonably well. You can apply some further refinement of ICA there, but if you're interested to develop another methods to remove eye blink, whether it's a wavelet or independent component analysis or any others, absolutely fine there. So I'm nothing against about that. But the way we talk about main application of wavelet here is not artifact processing, is primarily because there is no mother or daughter wavelet. There is only one wavelet. Everything is the same. We only have to fix the number of cycles there. That's it there, just to measure the time varying nature of the oscillations there. And now if you're having, depending on, suppose if you're using a wavelet to detect not just artifact, to detect some sharp transients there. Certain wavelets, depending on if you use the Dobbequist wavelets, certain order, they are much good at detecting kind of transients. Surely there is no one should stop you if you are having a specific aim, because as I mentioned, I cannot cover every possible methods there. Okay, so absolutely no one should stop you to explore any particular methods if you have a very specific aim there. Anything? Okay, so I'll see you then afternoon. Thank you.